Hello, hello. Aha, it works. Okay. Well, we are all in for a great treat tonight. Not only do we have a really interesting and fun topic, but we have a wonderful presenter. Kimberly Sasson is here tonight. Sasson, excuse me. She is a fellow master naturalist and an avid iNaturalist person with over 36,000 observations on iNaturalist. She's a certified entomology specialist, but most of all, she is a passionate gall hunter and researcher. And she fell in love with galls when she discovered them before COVID. And then when COVID was happening, she had all this time to dive into this research. And she's gone bonkers with it, and we're so excited to learn from her tonight. Professionally, she's a research assistant at BRIT, studying Colombian fern diversity. So she does specimen processing and she does DNA extraction, which sounds really cool. So her work bridges field operation and formal research. When I've been listening to Dana Wilson rave about her ever since she did a talk and walk out at Spring Creek Preserve, where on one leaf she found 4 species of galls, and one of which had been identified but never before named. So we're super excited to have Kimberly here tonight to tell us all about this fascinating part of nature.
Screen.
Okay, now try it. Yeah, there we go. Now you can hear me. Okay, excellent. So I am so happy that you're here. I have looked forward to this evening because I've never presented to North Texas Master Naturalist before. So I know some of you. I have met some of you before at the annual meeting, and I have done this presentation at the annual meeting. But it doesn't matter how many times I give it. It's one of those things that you will see something new every time. So if you have seen this presentation, I expect you to still be awake. Sam? I know you will. You'll be cheering me on, and I appreciate that. But also, for those of you who don't even know what a gall is, you'll learn tonight. And you're going to walk out so pumped and excited to go find a gall that I imagine you will be looking in the parking lot before you ever leave here. So I'm not going to be too surprised. So first of all, let's just start with what is a gall? These are some examples up here. A gall is an abnormality in the plant where the plant grows out a little bit as a result of an irritation from an insect, and that abnormality becomes like a nursery to an insect. And so what I want to make clear to you here as we go into this is that the gall itself, while there are many different shapes and sizes, that gall is completely plant tissue. It is not insect tissue. So if you take the gall, the strange looking abnormality, and you DNA extract it, you're only going to get plant tissue. You will not get any insect tissue unless you get the larvae itself inside. So this is something that the wasp or the insect that triggers it initiates with some chemicals, but does not form. It's the plant that forms it. So this is something that's really unique in the natural world. And common host plants. There are so many that I could not begin to list them. One of the most common would be oak trees. And you'll hear a lot of people talk about oak galls. And so we're going to focus on that in the beginning. But there are others, hackberry trees, roses, grapes. I see some of you shaking your heads, so you're familiar with some of these already, so that's wonderful. But also anything in the Asteraceae family, which would be sunflowers and things like that, the yellow flowers. And where can you find a gall? On any part of the plant, any part. Anywhere from the roots to the petioles, to the leaves, to the underneath the bark, anywhere. So that's what's so interesting, is any part of the plant can be infected, so to speak, with this gall. And there are different kinds, and you'll see that. So as we're looking through the different examples, keep your eyes open for where these galls are occurring, because some of them are in different places. And I just want to introduce you very briefly to the gall inducers. Inducers means the insect that is creating the gall, or the one that is using those chemicals to initiate that gall. So inducer is the one that's starting it. And these are some of the insects that do that. And these are the ones that we're going to talk about today. So I like to think of galls as the bed and breakfast of the natural world. The inducer is just one of the many insects that inhabit galls. On top of that, you also have inquilines, which are insects that will come along and lay an egg inside the gall that has nothing to do with the other insect. It just is taking up room. So it's like a roommate situation. They don't prey on that other larvae at all. They're just hanging out together. And they're both eating the inside of that gall as their nutrition. You also have parasitoids. There's a couple of examples over here on the right. Parasitoids are fun. I'll give you an example. A parasite is something which you're familiar with, will live on an animal and will not kill that animal. It will just feed off of it. A parasitoid will feed off of it to the animal's death. Okay? So in this case, a parasitoid is another wasp that comes along and lays an egg on the inducer animal, the animal that created the gall, and that wasp is using the larvae as its food source. Not the gall, just the larvae, until that larvae dies. And then you'll have a different wasp come out. So I have these wasps coming out, not at all what I expected whenever I was rearing them. And then we have something called hyperparasitoids, which this is really fun when you're trying to get the inducers to come out, and that's what you want to see. But you have parasitoids of the parasitoids, sometimes of the parasitoids, and you can do that like 5 different layers deep. So imagine how confusing that can be. So one of the interesting things for me is that galls are not just about a wasp. It could be anything. It's like a magic treasure box of you have no idea what's coming out of that gall. And so you can see some of the interesting wasps that came out that were not what I was expecting. I just think that these are cool still. And these were just sort of like wasps that I wasn't even interested in. So let's go back to the inducers and let's talk about these in more detail. We're going to start with the Cynipidae, which are the gall wasps. And these are very small wasps. They are smaller than a grain of rice. Think 2 to 3 millimeters. So super tiny. They do not sting. They do not bite. They barely fly. They are much more inclined to be kind of hopping around a little bit. So they will fly, but very short distances, and they're really not interested in it. I found out one day trying to take a picture of one, and it flew off, and I panicked. And I thought it was gone, and I came back the next day, and it just happened to be sitting right next to my camera. And I thought, well, if I'd known that, I wouldn't have panicked. It was so easy. But they do come back. One of the interesting things about gall wasps is they all look very similar. They all have this dark body, either brown, black, or a dark red color. They all have these really dark veins in their wings, and these sort of fuzzy little bodies, give or take some. But they're all generally built the same. If you were to look at them lined up together, thousands of them, it would be very difficult to tell them apart, even as a scientist with experience. So this is something that I find is exciting, because I don't need a degree to figure out which wasp I have, because the wasps are so confusing anyway, even the scientists can't do it well. So we look back at the galls themselves, because they're all different. And so we'll talk about those now. So as we look through these galls, I want you to pay attention to the forms and how different they are, and wonder what the function could possibly be and how that would change, because these are things we don't have answers to. For example, the one on the left is this short, furry little gall. Why? What function do the hairs have? That's a good question. We think maybe it makes it more difficult for parasitoids to inject their larvae into the gall. Could be any number of reasons, but that's one. I do have an example of that here on the table, so come see it. These are teeny, teeny, tiny, like really tiny, so you have to look for them very carefully. The one on the right, Androcus lustrans, is one of my favorites recently, just because it is so common for us. I am in the cross timbers area where we have post oaks everywhere. So it's hard to walk down the street without kicking post oak leaves somewhere. And these post oak leaves get this Androcus lustrans all over them abundantly. And a couple of years ago, we had a flush, I guess, if you will, and they were everywhere, even more so than usual. And a friend of mine sent me this picture, and he said, "What in the world is going on with my driveway?" And I said, "You have Androcus lustrans all over your driveway." And I confirmed with him that he did have a post oak tree right above his driveway, but the entire driveway is covered in red with these tiny little galls. And he was very concerned that he would suddenly have wasps just floating all over his house. And I said, no, don't worry about that. You're not going to have to worry about that. Even if they did all emerge at one time, what's more likely to happen is that they will fly away and you would never see them. Half of the galls are going to wash down the drain. The other half might get eaten by birds. And sure enough, the very next day, he came out and the birds were eating the galls. So they were pecking the larvae out of the galls. And so that was a fun thing to confirm. And he never did see the wasps. Cocosynops riley, on the left here, I think this one's really fun because it looks like it's got a little hat on it. We have maybe just recently come up with an idea of why it's got a hat. And the thought is that it encourages ants to carry it back to their nest. The ants are attracted to the hat part, which is somewhat enticing to it, and it will carry that whole gall off to its ant's nest, just like it does with other food sources, and then that gall will emerge later on from the ant nest. On the right, it looks just like leaves. But if you can imagine, this is actually a gall. So it's very tricky to start training your eyes to look for something that's different. But I knew when I saw this on a white oak that the leaves didn't look like that. So I thought, something's very weird here. And sure enough, the more I peeled into it, the more I found inside a gall with the wasp inside. So this is what it does to the leaves to make it look hidden. On the left, fuzzy dryer balls. I have nothing exciting to say about that. It's just fuzzy dryer balls. They're small and tiny, and they're really neat. They're fun to find. But the one on the right, I have fun with these. They are acorn plum galls, and you've just missed them. You might find some on the ground under red oaks. I see some of you shaking your head, so you've seen them. What I want to point out to you is that this one, if you can see, let's see right here. The acorn is the cup of the acorn right here, and the plum gall is attached to the cup, not to the stem of the tree. It's attached to the cup. So this huge gall, which is the size of a large marble, is attached to the cup of the acorn, not the tree itself. And it's one of the few that is attached to the cup that especially gets this large, which is rather unusual. Very pretty to find. I found these, collected a bunch of them, wanted to take pictures of the wasps, as I did with many. And I put these in a bag, which I'll tell you about later. And I found out the hard way you have to wait 3 years for these wasps to emerge. Needless to say, I never got a picture of those. On the left, this one is teeny tiny, smaller than a pea. Really, really small. But I love the little speckles of it. I think that that's just so cool. But amazing that you can just overlook it like nothing. It stands out in the picture, but when you're looking at the leaves, it's just so surprising to me how even now I can look at the leaves and just look right past them sometimes. And they're amazing, so different. On the right, you probably have seen these more than any others recently on live oaks. They're bigger, they're like marble size. And this is one that you may not see them in large clusters like this, but sometimes you will have a wasp that lays one egg and one gall forms, and then they'll fly away to another leaf and be gone. And so you will only find that one gall. And then other times you'll have them get a little carried away like this and lay hundreds of eggs, and hundreds of galls will pop up on one leaf. As Nancy mentioned, don't overlook the fact that you could see several different species on one leaf or on one plant. I can spend all day staring at one hackberry tree because they all have different galls. And they're so crazy. They're really, really neat, really cool to look at. We'll see some of those coming up. The oak flake gall. This is an example, again, of where they just sort of lay a whole bunch of eggs and scatter them around everywhere, really tiny, compared to the one on the right where they cluster them together. Androcus texanus is my gateway gall, as I like to call it. This is the one that I was out eye-natting, as I did, before COVID hit, and I came across this cluster on a leaf in January and picked it up and wondered what it could be. I knew it wasn't caterpillars, but I didn't really know what it was. I suspected it was wasps, but to me, wasps were paper wasps, right? Polistes. And so I took it home, I put it in a bag, I hung it on a tree, and I forgot about it for about 2 weeks, and I went out and checked it one day, and sure enough, there's all these tiny little wasps crawling around everywhere, and I think I went crazy. I think I photographed those wasps more than anything else. I think if you look at that one observation on iNaturalist, it's probably got the maximum number of photos. And then I went back and squeezed in a few more after that. So there is a way to do that. No, I won't be telling you how. Androcus biconicus is a neat one. It looks like cones on the plant. So it looks just like the plant itself, but it's one that I walked past it 100 times before I realized that's a gall. And I had to go look it up. And this is one that I spent some time studying post oaks just because they're so abundant where I'm at. And I said, I want to look at every gall on a post oak that I could possibly find. And I listed out all of them that I could find, and then I went to the research and I looked up all of those, and then I went and tried to find all the ones that I hadn't found before, and so that's when I came across this, and I went, aha, I've seen that. So that was a fun one to go back and try to find again. But again, you're looking for something that just doesn't quite look right, and that one didn't catch my attention the first time. The one on the right is commonly seen on bur oaks. It's usually very pretty colored, they call it a jewel oak for a reason. It gets these very vibrant colors as it changes and lignifies. So it's a neat one to come across. Amphiboleps confluenta. How many of you have seen this before? Yeah, everyone has seen this one. I brought a couple with me. This one is really large, but the fun thing about this one is the picture on the right, this right here is the wasp. It's smack in the middle, and it's got all of these radiating fibers that hold it in the middle of the gall. And the purpose for this is so that the parasitoid wasps cannot reach it when they come and try to lay an egg in that gall. So they would have to have a really long ovipositor to be able to do that. Believe it or not, there are wasps with really long ovipositors that can reach them. But for the most part, this one is not parasitized quite as often as some of the others are. But a big one, you'll see them a lot of times on the ground. Kids love them. They get very crunchy and crackly. So the cynipid life cycle is an interesting one. I won't spend too much time because it can be a master's degree program in itself. But what I want to say is that in the springtime, you have male and female wasps that mate, and then the females go and lay their eggs on leaves that are unfurling. And then in the summer, where we're at now, those leaves are growing, and the galls are growing now too. So as you can see, some of these leaves that I brought with me today are green, and these are ones that I picked this morning. So you can see that even in August, there are galls that are growing. And so it's a year-round exciting experience to be able to find galls. In the fall, the leaf galls will lignify and turn brown while the larvae are pupating inside. And then in the winter, the females will emerge in November and December and lay eggs in the rootlets of the tree just below the soil surface. So what I want to kind of point out here is that we have wasps that are male and female mating in the springtime and laying eggs on the leaves. And then we have wasps in the winter, which are females only, that are laying eggs on the ground of this particular gall. So you have males and females, and every one of these adults, the two different females and the male, can all look different. So I could have described 3 different wasps to science, plus a gall or 2. How confusing can that be? Now we got to try to wrap all of that up. So you can see what the scientists are working on now is trying to connect all of this, because now we have genetic testing and we can test all of these, and now we've come to realize, wait a minute, that's not a different gall, that's just a different generation of the same gall. So now we're starting to connect all of them together, and we're having to rewrite the natural history of some of these galls. Gall midges, again, really, really tiny. Everything is going to be tiny. I know that you've seen some of these. You probably don't realize that you've seen them. But I brought some with me today. The ones on the left, the Polystepha pilulei, they are fun. They distort the leaf. And if you ever get the chance to cut into one, the larvae inside is this super bright orange color. And what happens is when the rains come, or the moisture, and it softens the gall itself, and the top of the gall kind of puffs up and pops off, and then the larvae can fall to the ground and do its business in the dirt. So I think of them as little transformers, because they, like, the whole gall just kind of puffs up and spreads out. It's really pretty weird. On the right, hickory sombrero gall midge. This is just proof that scientists have a little sense of humor, because that's exactly what that looks like to me, is a sombrero. On the left, we have hairy pumpkins. I don't know who came up with the rosettes, but yeah, these are hairy pumpkins. These are some of the ones on hackberries. And again, these are the ones that you can find multiples of them on one leaf. So it's easy, just keep looking. On the left, this is your gall right here, this little fold. That's all it is just a fold. And I brought one with me today. I was lucky enough to find one. There's also one with me that is where it's along the veins, the midrib of the leaf, and it's like a little crust kind of gall. So it's interesting as well. But they can be very inconspicuous. Or on the right, I think of these as little bishop's hats. They're kind of interesting. I don't know, columnar gall midges. They missed the mark on this one. Acorns, I don't know, they could have called it anything other than columnar. He got tired, I think. And then gall aphids, psyllids, and phylloxera. So I'd love to tell you more about aphids and psyllids and phylloxera and give you this very technical explanation about insects. I'm just going to say they're really small. They are plant-eating insects, and the galls that you find are just as unique as the wasps and the midges. You can see on the left, this is a hackberry gall. It makes stars. It looks like stars have come out of the leaf. On the right, this is what we call a nipple gall. One of the things that I used to tell people early on is that you should take pictures of both the top and the bottom of the leaf. This is a perfect example of that, because on the top of the leaf, it has this pit, but on the bottom of the leaf, it has this ball, this nipple. And so it looks entirely different. So if I were to take a picture of the top and show somebody, they might not recognize that because they're used to seeing on the bottom side. So it helps to take pictures of both sides, but then I found out that it's a lot to do with the hackberry galls, and not all galls are like that. So it's not as much on the oak trees and things like that, so it's not as necessary, but definitely on the hackberries it's helpful. On cottonwoods and poplars, aphids have a tendency to warp the leaves. So you'll see, especially along the midrib, it will be bent. And that's what's happening here on the left, is it's just bent this. And if you cut into it, you'll see something like this. And there's the adult right there with all the little babies. Moths also will make galls in plants. Not as often, but they do. And we only have a couple of examples, but this is some fun stuff, I think, because I am also a mother at heart. And so I love to see the moths doing what I love to do, is hunting galls. And so this is a grape leaf here, and you can see the galls on it. What's really interesting about this is, here's the caterpillar, the larvae. They cut the top of the leaf off whenever they're done. So they just cut the top part of the leaf off, and they wrap themselves up in it, and then they fall to the ground. And they will stay in the ground for a season or a year until they're ready to pupate and then come out as a moth. I don't have a picture of the actual moth on this one, but I thought that the larva was a little bit more interesting. But I measured one day, this is how twisted I am, that they can crawl 2 millimeters in a pool. Isn't that interesting?
Cool.
Yeah.
2 millimeters.
2 millimeters every time he pulls.
Oh. Is that like 45 minutes or something?
Yeah, it did take them a while. They move actually pretty fast. These do. I guess they don't like being out on a table. And then on the right, false indigo, if you are familiar with that plant, it's one of my favorite plants. I just happened to come across this bulge in the stem and cut into it and found out that it was a caterpillar. And then I raised it and got this moth out of it. And so that was pretty interesting. So we don't have a lot of plants that have moth galls, but there are a few. And then the gall mites, the areophyids. These are something that people have seen quite a bit now. They've been around, at least in this area, for many years. But I remember 10 years ago now texting Sam and saying, "What the heck is this?" And we had no idea at that point. And it's been one that keeps coming up every City Nature Challenge. They're out in April, and people take pictures of them. And to this day, it's still undescribed because there are no scientists studying areophyid mites in Texas that have funding, I guess, to work on this. So we're patiently waiting for anyone that will describe this one because we know that this was a unique one. So no, don't look at me. I'm not doing this one. That one's too hard. So far, I've only seen it in Texas. So that's a good question. I can't say. I don't spend a lot of time looking at observations outside of Texas, but yeah, that's a good question. On the right, even this little flake of stuff is considered a gall. So mites are causing that flake. And then they also will make little balls. If they're really small like this and sort of speckled, pimply looking, you can pretty much guarantee that that's a mite of some kind, especially if it's on an herbaceous plant. The one on the left is ragweed, western ragweed, and the one on the right is poison ivy. So you might have seen that while you were trying to avoid the poison ivy, that even the poison ivy gets infected. So, So why look for galls? This is kind of a silly question because, you know, you're going to love them and you're going to find them so interesting. And they're really amazing to look at. So why not look for them? But they're fascinating, and they come in so many different shapes and sizes, and each one of those is different. So what I want to leave you with at this point is that each gall is unique to a particular species of plant. Each wasp is laying its egg on a red oak or a white oak, but it never picks both. It is very specific to that. And so if you show me a gall, I can tell you what tree it is without even seeing the leaf, because I've studied those galls so well, I can relate them back to the trees. So that's something that is interesting. We can tell the galls by the host plant that they're on. So it's important to note the host plants. And also that it takes no scientific knowledge to study these. This is something that you can go out with a camera, take a picture, and I'm going to show you how to look up an ID on them very simply. And so this is the fun part. But as I said before, I went out today, I brought back a sample of galls that I collected within under 30 minutes on a variety of different trees, on oaks and hackberries, and they're abundant, and it's August in Texas. So there's no reason to not be looking for galls year-round, because they're out there. Many of the species are unknown. As a matter of fact, 1/3 of the galls in Texas have not been described to science before. 1/3. That's of the galls that we know of. There's a whole other set of galls that we don't even know about. So the more you're out there, the more we find, the more we need to describe. And we have researchers now that we didn't have 3 years ago that are interested in what we're finding. And so they're anxious for us to find new species and to work with them on it. And then there's many of the natural histories that are unknown. For example, when you describe a new gall wasp now, they want you to close the loop, so to speak, and that is to describe all of the adults, if you can, all of the generations, including the gall, and not just the one that you happen to find. So the idea is to get as much history as you can when you're describing it. And there's a lot that we just don't know. Sometimes we have to publish with what we do know. Speaking of publishing, I have the pleasure of being friends with Bob O'Kennon, who is a research associate at the Botanical Research Institute of Texas at the Fort Worth Botanic Gardens, which is a mouthful. He is a naturalist at heart, not just a botanist, and he was out looking for galls. He got very interested in the galls one day and just went wild, and would call me every couple of days and say, I found something new, I found something new. And so I was constantly meeting up with him. And one Thanksgiving, he called me and said, I found something new. And I said, actually, I think you did. I think you did find something new. So I went out and met with him and looked at this plant. And what was so interesting about it to me, what caught my attention, is you can't really tell, but here's the galls. There's multiple here. But what's unique about this one is that on the underside of the leaf, there are galls on it as well. And it's unusual for that to happen. Most of the time, on leaf gall, it will be either the top or the bottom, but very rarely both. And this is one that they're abundant on top and bottom, and there's a lot of them. And you can see my little friend popping his head out. I have lots of pictures. I have thousands of these wasps. So I said to Bob, he's not quite this crazy in person. Well, maybe he is. But I said to him this day, can we recreate that photo? Because we didn't have our cameras with us, so we recreated it. So this is him getting excited that we had found an undescribed species. And so we talked about it, and I reached out to an iNaturalist colleague, and he works at the Smithsonian, and he agreed to help us describe it for science. And so we were able to describe, and we were able to name this new wasp Druon lacei. So we named it after the oak tree that we found it on, the lacy oak, which coincidentally is not a common oak around here. It's one that got brought into the Fort Worth Botanic Garden quite abundantly. We have probably 50 or 60 different lacy oaks, but it's more from central Texas and down into South Texas and Mexico. But this gall has only been seen at the Fort Worth Botanic Garden. So we have yet to find any other galls. Even though the tree is abundant in other places, we still have not found this gall anywhere else. So we've got our eyes open looking for that. So I'm going to go quickly through some of these slides because you may or may not be interested in collecting and rearing galls, but I get a lot of questions about how I do that. And so I want to show you some of this. Photographing galls for identification. You're all required to be on iNaturalist, right? Okay. I said that right, required. So you take pictures of the gall on the host plant so that if you can't tell what the host plant is, somebody else can tell what it is. I can tell that this is a hackberry. And so I can see the gall and the size of it on the leaf. And then I take a close-up of it. If I happen to have a knife or something like that, or a pair of scissors or clippers, I will often cut that gall in half to see the inside of it. And for me, that shows me the arrangement of the chamber inside. This is not necessarily something that you need to do anymore. Again, something that we used to do a lot back when we didn't know a lot of the galls. And now we know so many of them. But what's going to happen is you're going to assume that everything that you find is known. And then you're going to find one that is new to science. And you're not going to do that. And then you're going to regret not doing it. So just do it. So cut it in half lengthwise so that you can see where that chamber is and where the larvae is. Because there's another gall that looks very similar to this, where the bottom flares out just a little bit more, and the chamber goes all the way to the bottom. But I did not know that until I cut into them. So take pictures, and then go to gallformers.org. And this is a website that we put together around the time of COVID, a little bit after that. And the whole point of this website is for you to be able to compare your pictures with pictures of other galls. So it is super easy. You don't have to know scientific terms or anything like that. If you want to narrow it down by where it's located on the leaf, on the upper surface of the leaf, on a vein, whatever, you can do that. But you can just scroll through the pictures, and you can see here, my gall just happened to come up in the very first ones that I looked at. I also want to point out, for those of you that are wondering, we also have non-gall things that often get mistaken for galls that are on there as well. So don't get confused by that if you do run into it. When I'm collecting for rearing, I collect a whole bunch of galls in one bag from one tree. So I label my bags this way. And if I'm collecting the leaf, I will collect the whole leaf with the petiole. If I'm collecting from a stem, I'll collect a couple of inches above and below the stem. But these leaves are not like caterpillars needing fresh milkweed or something like that. They're not going to wilt at that point. If you collect them too early, then the gall is not going to survive anyway. But if you collect them later in the fall time, then you will have better luck with them making it through pupation because the leaf will be more mature. So it's a great idea to wait until the fall to start collecting them. I usually do this in October and November. But I go through and collect a bunch of them in a bag, and then I go home and take pictures and sort them all out into little miniature mesh bags, into little wedding favor bags. And I put them in there, and this is where I wait for the wasps to come out. I label them with UV markers so that they don't wash off. I put that bag outside and in a bigger container, and I leave it so that the galls are exposed to the natural elements. I want them to be exposed to the weather and the humidity and all of that we normally get as much as humanly possible, so that they're emerging at the appropriate time and with as little human influence as can be. Here, if you look up here, are the wasps. And this happens to be the one that took 3 years. These are not the inducers, these are parasitoids. So these are the ones that would fake me out, and I would get so frustrated. But there's value in parasitoids as well, because there's a whole bunch of researchers that are studying just parasitoid wasps. So there's value in those. I don't want to disregard that. If you happen to have a spare 9-year-old laying around, they are really, really good at collecting wasps, let me tell you. I take a little vial of alcohol and a toothbrush or a paintbrush, either one, and you can scoop that wasp up on the brush, super easy, and just tap it into the water. Like I said, they don't fly very well. They hardly even jump to amount to anything. So I just kind of dump them out on a piece of paper, and you can see my niece here collecting them and sticking them in the alcohol. And we preserve it in 95% alcohol for genetic testing. They will do all sorts of things. They will chop them up into different pieces and do SEM images of the antenna and the head and the legs and the wings. And keep in mind, some of these adults, while the scientists have to describe them down to the hairs on the wings, that's not something I can see with my eye or with a camera even. But they can see that with SEM images, and so they do take those wasps, and so they need several of them to be able to do that. And then reach out to gall formers for a contact if you're interested in that. And I want to leave you today with just a little fun fact, and that is that the Declaration of Independence was signed with gall ink, in case you didn't know that. And you are welcome to Google the recipe for gall ink. It's a lot of fun. It has a little bit of a variety of recipes online, so there's no one tried and true. They all work, but they're fun to try and paint a picture with. And now it's your turn. Now it's your turn. Go out and find a gall. And I have given you my contact information, both my email and my iNaturalist handle, so you can tag me, you can email me. I even get emails at Brit saying, look, I found a gall. And I'm like, this is wonderful. I would much rather get a gall message than a work message. So I invite you to email me and to tell me what you found, because that is a much better way to spend my day than working. So if you have questions, I'm happy to answer them. Yes, Nancy. We need a microphone.
Just a second. So, Nancy, you're my volunteer. Thank you so much. You can go first, and then we're going to alternate between people in person and people online.
So I just had 2 follow-up questions. Mm-hmm. One was, it's just laying eggs is the irritant to cause the gall to form?
Yes, it's laying eggs, and there's a chemical that we believe is released at the same time as the egg. And that chemical mixing with the plant is what causes the gall to form. It's just, we don't know enough about why wasp A has a particular chemical that causes the gall in this tree to be round and spotted and form on the leaf midrib, as opposed to on the trunk underneath, the bark on another one, even though they're entirely separate species. There's just so much variety, and why are those chemicals different? We don't know. So there's a lot that we don't know about those chemicals yet that's still being studied.
And my other question was, do you have a picture of the wasp that has an ovipositor long enough to...
I don't. But I will say the ovipositor, the ones that I've seen, the ovipositor is longer than the wasp itself. Yes. So, right? Anybody? Yeah, like really, really long. So, yes, yeah. So look them up on iNaturalist.
Thank you.
We have a couple of people online asking the same question. If they cut the leaf off, if they find a gall on a plant leaf and cut the leaf off, the leaf will die. Why does that impact the gall?
The gall is growing at a different rate than the leaf. The leaf is unfurling at one rate. The gall is growing at a different rate, and the gall itself is a very thick texture. If you cut into them, you can usually see, especially on oak leaves, they're often really thick and spongy. The insect is eating the inside of the gall. So I don't have an understanding of why. If you take a leaf in August, most likely that gall is not mature enough to produce a mature wasp. So it will not last long enough to pupate that way. But if you wait until October or November, it's been long enough on the leaf with the nutrients that it will. So that's why I recommend waiting until October or November.
I think you said you counted all of the gall species on a single tree?
Yes, I did.
How many was that?
Oh, gosh. It was a post oak tree, and I don't know that I came up with an actual number. It was well over 100 on just the post oak. But I had a bunch that I could not find here. And one of the things that I discovered was if I went to a different county, that those wasps were there on the post oak in a different county. So it was really interesting to travel and see galls that I did not find here that were more common in other places. So everywhere I go, I look for galls. Everywhere.
So Beth asks, is there any evidence of galls within the fossil record?
Yes, there is. I don't have a lot of details about it, but I can tell you that there have been galls found in the fossil record. So I'll leave you there to do your own research on that one.
Do you know if the hyperparasitoids are targeting a specific species of parasitoid, or are they just targeting whatever is inhabiting the gall at that time?
Both. So there are some that are very specific to a particular gall, and there are some that are generalists. So we do have both.
But so if they're parasitizing another parasite,
Mm-hmm.
Can they tell what parasite is already in there?
No, I don't think so. Yeah, so that's a good question. Yeah, unless they follow it around, which I guess is possible.
Sue Yost asks, when you cut into a gall, does the insect inside die off because it's exposed?
Yes. So there are creative ways that you can put the gall back together, depending on the type of gall it is, and tape it up, glue it back together. There are ways to do that that have been effective, but I will say there is value in collecting the larvae for genetic testing as well and for describing them, in addition to the adults, the galls themselves, etc. So when it's possible, collect more than just one. So collect multiples for that reason.
Kimberly.
Yes.
Great presentation.
Thank you, Sam.
Would you brag a little bit? Who took those photos?
I took all of them.
Yes, you did. Yes, you
Except
did.
for a couple. There were two. I didn't take the driveway photo, and I didn't take the grapevine, which
And how did you get such good photos?
practice. It is very hard to take pictures of something that is only 2 millimeters long. So it's a very expensive camera. It's an Olympus. I think I probably spent around $2,500 on my camera equipment. Plus, I have a light box, and I have microscopes and all sorts of stuff that I just never use, and I have a whole lab at my house just for wasps. And I have had to stop using it because my husband put his foot down and said, "No more bugs in the house." He's convinced that I'm the cause of whatever we find in the house. And I can only say I didn't do it so many times, although I probably did.
So, Kimberly, a question for you, though, too.
Yes.
So you did talk a bit about the plants, which is super important.
Mm-hmm.
But I'm kind of curious. You've inspired us to go hunt galls. Do we focus on monocots, dicots? Do we look for perennials, annuals? Do we look for mosses, ferns, junipers? What group of plants do you think has the biggest proportion of galls?
So the biggest proportion would be oak trees, hickories and pecans, hackberries, and asteraceae. Anything in the asteraceae family, like sunflowers, anything in that sunflower family, or compositae, if you're familiar with that word instead. Those are the most common. I would say don't discount anything if you are advanced and you've looked at all of these plants before and you feel like you've seen everything there is to see. I have found a gall on everything I could imagine except a redbud, an eastern redbud. That's the one plant I cannot find a gall on. And so I'm frustrated with that. I'm still looking, but
Perennial or annual?
both, perennial and annual. I have pulled up some perennials by the root to get them out of the way for something that I was looking for, and found the galls on the roots of the perennial. Yeah. So it's just, yeah, anything. If there's a plant, there's a gall for it. And it's just a matter of being in the right place at the right time, and finding that. And then I would encourage you not to assume that it is known. Treat everything like it's an unknown, and then go back and go, oh, darn, they already have a name for this one. Because there are so many that are unknown. And one thing that we do on iNaturalist is, if it's undescribed, we have a code that we put with it called a gall formers code. And so we code all of those that are undescribed, so that if there is ever a name that is assigned to it, then we can go back and put that name on all of them quickly. So you can see that group, the ones that look like Captain Crunchberry cereal, those little red tiny balls. I can click on that gall formers group name and see all of those, and see how many there are and where they've been seen. Map them all.
So the gall is all plant matter.
Yes.
Why is it that the same plant will make different shaped galls for different insects?
That's the question. The chemicals is the best that we know at this point. The chemical reaction of the specific insect to that specific plant. Yes.
These are such tiny things that I'm assuming they don't harm the host.
They do not. That's a great question. There are very rare instances where a gall will cause damage to the host plant. And you'll have a lot of people, especially with live oak trees, mealy oak galls. I know that's something that people are familiar with. Because they're unsightly, they're big, and they are ubiquitous, I guess. They're just everywhere. They just get all over every live oak tree. And people often ask, do I need to do something to the tree? Do I need to have the tree treated? And galls are generally more cosmetic issues. They're not damaging the tree itself. They're walled off from the rest of the tree. They're not sucking nutrients from that tree to the point that it's causing any damage whatsoever. Now, having said that, if you go look up grape leaves and galls, there's a whole history of alcohol and wine and how galls affected that industry. So it can happen, but very rarely. It's not a common thing. Yes.
We're still online here.
Okay.
Are galls found worldwide?
Yes. I don't know. I'm sure that there are places that they aren't found, Antarctica or something, but I don't know the details of that. But to my knowledge, they are found in all major continents, yes.
Is there any benefit to the tree for having the gall?
Oh, that's a good question. Benefits? I don't know about benefits. I do know that the galls will attract ants. Some galls will produce a honeydew, and that will attract ants and wasps. So those could be clearing off predators. As let's say a paper wasp comes along and eats the honeydew off of a gall, and then happens to find a caterpillar right there next to it that's munching the leaves and carries that caterpillar off to its nest. That's possible. That's a possible benefit, but generally there's no benefit to the tree of having a gall. No. Online?
Yes. Beth asks, she says, I found gall midges on a cultivated prairie acacia in my yard. It was the first of its kind to go on the gall former site, and they created a code for it.
Yes.
It makes me wonder if these were present at the nursery, or did the bug find its way to my yard randomly?
Oh, that's a good question. Very possibly either one. I would say that's kind of the similar situation with what we have with the lacy oak gall that we described. The plants were brought in and planted at the garden, and we don't know where that wasp came from. We make the assumption that the wasp came with the plant because we don't have that plant here otherwise. So I'm inclined to say that it probably came from the nursery, from the dirt and the native soil that came with the plant. That these wasps and midges have a tendency to stick to one plant and not wander too far from it, or they will wander very close if there are similar species right next to it. But as far as a wasp or a midge from this area finding your plant, if we have not seen that midge before in this area, then I would say probably not. Probably from the garden center where the plant came from originally.
You may have already answered my question, but so the wasps and midges, they're specific for a specific kind of plant.
Yes.
So I was thinking, are there any cases where the wasps or midges will go to more than one kind of plant? And if they did, would the gall look the same or would it look different?
There are instances where the gall wasp will go to a different oak tree in the same family. For example, it will go to only white oaks, but it will do various white oaks. It could do a post oak and a sand post oak because they're so closely related. But it probably won't do a white oak and a post oak because they're not close enough related, even though they're both white oaks. So there are degrees to which these insects will adapt to some extent. So when you look on gall formers, when you're looking up one particular wasp, you may find that there are several hosts that are listed. And that's because they have been found on several plants, but usually they're very closely related or hybridizing type plants, that sort of thing. But it's usually on oaks and things like that, not jumping from a white oak to a red oak, never. Not that I am aware of. I shouldn't say never, but not that I'm aware of. If it goes from a post oak to a sand post oak, the gall looks the same. The gall may have different sizes, but generally the features of the gall are the same. Enough that we might question, is it the same one? But then when we look at the wasp itself and the features of the gall, when we cut into it and all of that, when we really start to look at it, it all turns out to be the same. Yeah, good question.
Are root knot nematodes considered
Where am
a
I
gall former?
Oh, was that you?
Yes.
Sorry. Root knot nematodes. Yes, nematodes can be gall forming. Yes.
So the female, the second adult phase of the oak gall wasps, the female phase lays eggs in the soil. Are we aware of what the larval food source is in the soil before it becomes the asexual?
It's the root. And I should clarify, that was for one particular type of gall. So the female form could be not necessarily a root. It could be something, it could be a different location on the plant. So different galls have different locations for their asexual and their sexual generations. That particular instance, it goes to a root, and so they're building galls. So they're building another gall once they get there in the root, and so you can pull up the roots and actually see the gall. It looks like a potato in that one. It looks like a potato on the roots of a live oak. Live oak is a good one. So if you're ever around a live oak, dig it up, and especially in the wintertime, see what you find. Any more online? Okay, great.
For the DNA testing, like who is doing that and how does that work? Do they just take anything and anything and then try and get the information in the right spot, or is there like a wish list and only those things get sent in, or how does that work?
Different labs do different things. With gall formers, we have several labs that are studying gall wasps and the parasitoids that go with them, and they all have a wish list now. It used to be that they would kind of take whatever and then decide, and that's why a lot didn't get described before. So now they are describing more of them and putting more of the natural histories together and doing more DNA studies on these wasps together because they're looking at the whole complete picture of one family or something like that. So a lot of that now is done by laboratory and by funding. So as you can imagine, academics require funding to do their work, and so funding requires grants, and grants requires the National Science Foundation and other such organizations, and that's been very difficult to come by lately. So it's been a challenge, but they are out there still working off of their wish list. And so now we have a list of things that they want to see, but if there's anything new that kind of fits with it, they're willing to take it. They're excited about those too. And Texas is just not a very popular area to have been studied. We do have a lab at Rice University that is studying live oak galls primarily, but they're looking at other things too now. But we just have not had a lot, especially if you go towards the west. If you're out in the panhandle at all, there's so many galls out there on the Havard oaks that have just never been described. I have a list of them sitting at the Smithsonian right now that are just waiting for somebody to come along and describe them because they've been there so long. And so I just need to get them back and get them into rotation with the labs that are studying now. And same with the parasitoids too. There's a lot of people that are very interested in particular parasitoids and what they are parasitizing and what they're related to and what they look like, and having DNA studies done on those as well. Yeah, the genetics are fun. Okay, well, I appreciate your attention for tonight. I hope you go out and find something exciting, and please do message me or tag me on iNaturalist. Let me know what you found. Before you walk out, I have some examples over here, and I'll show you some of those as you are headed out the door. Please do stop and take a look at them so you have some idea of what you're looking for. Thank you.
I saw you all whispering to each other. Let's raise galls. Let's raise galls.